a second. So we're looking at cardiac progenitors that become at least vascular, endothelial, smooth muscle, and myocyte lineages. Next slide. So these are the alternative methods in cardiac regenerative medicine. These are the new breakthroughs in taking a fibroblast and transdifferentiating and converting them into another cell type. They're expressed by the viruses that we put into the cells, and in the top figure, it's that's what fibroblasts look like. Well, these fibroblasts, and then when we add Ets2, we turn them into little BBs. We turn them into cells that are just like stem cells. In fact, they activate a number of the stem cell markers, but they're not truly stem cells. If you remove Ets2, it eventually comes back and the cells go into some sort of intermediate morphology. But these cells have some of the aspects of stemness, but they're not true stem cells. And then if we take Mesp1, which has been cited as the key factor, the uber factor that drives all of cardiogenesis, if you put them into fibroblast, they don't do anything. They just sit there. But if you look at some of the genes that can activate, they activate some of the pathway genes that are involved in early mesoderm, and they mimic mostly cells that are going into the endothelial pathway. And if you sort these cells using a machine called the FACS sorter, facilitated activated cell sorter, you can see the separation of cells, and they're mostly showing a cell surface that is common to endothelial cells. It expresses KDR, and that's sometimes called CD301, 309, and PCAM, which is CD31. So these cells are enriched in that kind of activity. And then next slide. What we do is we make a reporter, and we want to be able to follow these cells as they mature into become myocytes. And the reporter is from a gene called NKX25, which I worked on for many years. And we know where the regulatory regions are that are necessary and activated in early embryonic stem cells that become hard. And we took those sequences and we cloned them adjacent to a reporter. So every time this promoter gets activated, it puts out a red color. And the dye is called TD tomato. It's a protein, and it gives a chemiluminescence. And what you can see in panel B is, and C, is that's a region we put it into embryonic stem cells. We can make them beat by hanging them and forming embryonic bodies. And what you see is beating region, and that's where the concentration of red stain is. We can cell sort them to get the red containing cells out.